Hi, good morning, everybody. Let's see, I'm not as tall as David. There we go. And I just popped off to the restroom. So I'm ready for whatever and however many questions come after this. So Helen already introduced me. I know many of you. I'm Christine Coquery. I'm an executive director in the Neurology Clinical Development Group at Ionis, and I lead our program in MECP2 Duplication syndrome. So thank you to Helen and the other organizers for giving me an opportunity to be here today to give you an update on the program, where we're at across all of our clinical trials. And then for those of you who maybe aren't as aware or familiar with Ionis and our technology platform, I'm gonna do a little bit of background on that. But my goal is to not sit up here or stand up here. I won't sit, I promise. Stand up here and just talk at you. I'm here to answer questions. Paul is here to answer questions. Courtney, who just disappeared, there she is over there, is here to answer questions. And that's really our goal today. A huge thanks to many of you who submitted questions ahead of time. We've done our best to incorporate the answers to those questions into our talks. But if we don't hit on something, call us out. Ask us whatever you have on your mind. We're here to help and we're here to answer questions. So these are my disclosures. So importantly, the safety and efficacy of medicines in clinical development, including Ion 440, have not been established or approved for use by any health authority. So the information contained within this presentation is not medical advice, and decisions about care should be directed to and discussed with your healthcare provider. Okay, so by show of hands, how many of you are familiar with Ionis and our ASO technology? Okay, a lot of you, great. So I'm not going to spend a huge amount of time then talking about it, but I do want to provide some background for those of you who maybe this is your first time hearing about our company and hearing about our technology platform. It's important for understanding how Ion 440 might work in individuals with MDS. So Ionis was founded over 35 years ago. We remain a leading innovator in RNA-targeted medicine, and in fact, 7 of those have been approved. So we're highlighting here all of the medicines. These are all approved in the US. I will note that many of these medicines are also approved in the UK and Europe. And I'm going to talk a bit about our neurology franchise. So at Ionis, we have interests in several different types of diseases. We have a very strong foundation in neurology. So we have 3 approved medicines in neurology, and we have 12 investigational compounds that are either in clinical development or within the next 6 months will be going into clinical development. 8 of these are wholly owned. So Ionis has a model where many of our drugs are developed internally in-house by Ionis, and many of the drugs that we developed are partnered, which means they're developed by another company. So 8 that are currently wholly owned and being developed by Ionis, and that includes Ion 440 for the treatment of MECP2 duplication syndrome. Now, at our company, our neurology franchise has 4 pillars. We have a heavy emphasis on pediatric neurology. So you can, the laser. So you can see our program in Alexander's disease, Angelman syndrome, PMD, I won't even pretend to pronounce it, MECP2, and then we recently announced our program in Dravet syndrome, all within pediatric neurology. So we have a strong foundation, a strong knowledge base, and a strong expertise in effectively developing medicines in pediatric neurology. We also have an interest in dementia, as well as motor disease, and movement disorders. So you see that peppered into our franchise here. So in order to understand how ASOs like Ion 440 work, it's important to understand how the information in a gene like MECP2 gets made into something that actually functions in our body. So you heard a little bit about this from Davut, but this concept of moving from DNA all the way to, and I don't have it listed here, protein, but protein are the functional units of our body. So anything that is in our genetic code needs to be made ultimately in that protein for it to have a function. And this happens through a process where DNA gets turned into mRNA, messenger RNA. And why is this important? Because ASOs act on RNA. So depending on the design, they can increase the protein, they can decrease the protein, or they can even change the protein that is produced, depending on the target of the ASO and the design of the ASO. They're highly specific and they're titratable. We'll talk about why that's important in a dose-sensitive gene like MECP2. So ASOs, or antisense oligonucleotides, like ion 440, are taken up by the cell through this process where they bind to a very specific target sequence. So ion 440 is designed to bind to the pre-mRNA of MECP2. And when it does that, it recruits, which just means it calls another protein to help it out. And in this case, it's RNase H1, which is an enzyme. And enzymes have a very specific job. They cleave things. And so RNase H1 comes in, it cleaves this complex, and in the case of ion 440, it's designed to lower MECP2 levels. Now, importantly, we are giving very specific amounts of ion 440. Why? Because we only want to reduce some MECP2. We're looking to achieve a physiologically relevant and still functioning level of MECP2 because, again, it is a dose-sensitive gene. You heard Dr. Pelevin talk about Rett syndrome, which is a loss of function of MECP2, and in MECP2 duplication syndrome, as the name suggests, we have too much MECP2. We're looking to get the right amount of MECP2 for homeostatic or normal functioning, and that's the goal here. I will also highlight ion 440 is designed to be a treatment and not a cure. You will have to continue to take ion 440 or another ASO treatment throughout the lifespan. So, how do we administer our ASOs for our neurological conditions? Now, what's important is we want as much of the drug to get into the site of the body where we need it to act, which is the brain. Now, the brain has this very clever little barrier around it, and that's very important because it's there and designed to help protect the brain from harmful substances. It does, however, present a problem when you're trying to get drugs into the brain. So we circumvent that issue by injecting the drug directly into the space that surrounds the spinal cord and the brain. We do that with a very common procedure. It's called a lumbar puncture. Colloquially, it can be called a spinal tap. And what that's done normally, under normal conditions, is if somebody is sick and there's something going on with the brain, then you'll do this procedure, perform the lumbar puncture, you'll collect cerebrospinal fluid, or CSF, and that's going to tell you a lot about what's going on in the brain. Now, we can leverage that to also administer our medications. So before this procedure ever happens, we do a couple of things. We're going to use a topical numbing agent. So that's something that can go on the skin itself to make the injection less uncomfortable, so less painful. This procedure requires you to stay relatively still. As you might imagine, we're going into the space that surrounds the spinal cord. And so for individuals where it may be challenging to hold still for the procedure, we can use general anesthesia, we can use conscious sedation, again, to make the procedure more comfortable and safer for the individual who's undergoing it. So how does this work? I've got a series of pictographs to kind of go through it. During an LP, a fine needle is inserted into the lower back. It goes in between the vertebrae, which are the bones that make up the spine. And we pull out CSF, which is the fluid that surrounds the spinal cord and the brain. Now, in the context of the clinical trial, this is very important. We can pull CSF. We can use it to measure biomarkers. Davut talked at length about potential biomarkers that either tell us something about the disease itself or, in the context of the trial, the disease progression. So as the disease changes with a potential intervention, what does that look like? We can also look at that CSF to measure the amount of the drug, and it can tell us that the drug is getting where it needs to go and potentially that it's doing what we expect it to do. And that's why that's so valuable. And then after the CSF is collected, you can inject the drug, ion 440, in the exact same space. So it's not a separate injection. It's all done through that initial injection. So that's just some basic understanding about Ionis, about ion 440, and about how ion 440 is administered. So I'm going to shift gears now and give you what I know everybody is here to learn about, which is what's the status? Where are we at with our program in MECP2 duplication syndrome? So we'll start back in 2013, which is when this program started, and later today we're going to hear a nice talk about the drug development process. I think Davut did a great job highlighting just the biomarker study, the biomarkers alone, how many years it took to get that data out and published. It's a long process. And so in 2013, our MECP2 duplication syndrome program started with an initial collaboration with Dr. Huda Zogby. Subsequent to that, Ionis screened thousands of ASOs to identify Ion 440 as a lead candidate. And then we took Ion 440 into safety tox studies to ensure that it was safe and tolerable in an animal model before we introduced it to a human. So in parallel to all that work was a lot of work that was being done to identify biomarkers. So the Baylor Clinical Trial Readiness Study, the natural history study that we'll talk about as well today, as well as other collaborators all over the world who are looking to find both soluble, so that means biomarkers that we can detect in the blood or the CSF, as well as non-soluble biomarkers. So some of the electrophysiology work that Davut talked about and you'll hear more about. And we were doing that so that we could try to monitor for changes in MECP2 and also look for ways that we could monitor for changes in the disease. After we did all of that, we worked with experts in the field of MECP2 duplication syndrome as well as Rett syndrome, and we worked with global health authorities and global patient advocacy leaders to design the ATTUNE Trial, and in October of 2024, we started the ATTUNE Trial. So here it is. This is the trial design. I'm going to talk a little bit about the trial design, and then we'll talk about the status of the trial. So the ATTUNE Trial is considered a phase 1 through 2. So phase 1 is a first-in-human trial, and that's how this is when you first start in humans, you go into a phase 1 trial. Now, we don't go into healthy volunteers with a lot of our neurological conditions just because of the way that the drug is administered and because of the importance of MECP2 in this case for normal neurological functioning. So we went straight into the patient population, and we also included key exploratory efficacy objectives. So going into the patient population and looking at potential early indicators of efficacy is something that you would expect to see in a phase 2 trial. And so again, a phase 1 through 2. Now, this study is divided into 2 parts. The first part is a double-blind, sham-controlled, multiple ascending dose study. It's a mouthful, so let's unpack it a little bit and talk about what all these different parts actually mean. Double-blind means Ionis, the investigator, and the participant and their family will not know whether or not the participant is receiving Ion 440 or sham until the end of the study. And that's the full end of the study, which can be frustrating potentially to individuals who enter the study at the very beginning. In order to maintain the integrity of the study, we actually have to make sure that every single participant, down to the last individual in the last cohort at the highest dose, has completed part 1 of the study before we can unblind the study. And when we do that, we'll all find out who was receiving Ion 440 and who was receiving the sham control. Now, sham control, what does that mean? That means, and as I alluded to with the whole blinded nature of it, that some individuals will receive Ion 440 and some individuals will not receive Ion 440 in part 1 of the study, but they'll undergo all the same procedures, and that helps us maintain that blind. Now, we are evaluating 3 dose levels of Ion 440, and that's to ensure that we understand and can take the best dose into a potential efficacy trial if the results from the ATTUNE study warrant additional development of Ion 440. And we're doing that in 2 separate age groups. So we're starting with an older patient population, 8 years and older, and then we'll evaluate that same dose level in a younger patient population, age 2 to 7 years old. Again, all of this is to help optimize and design a future efficacy trial, and we'll talk about the goals and objectives of this trial as well. Now, part 1 is potentially 42 weeks in duration. That includes the screening period, the treatment period, and then the safety follow-up period. Now, individuals who complete part 1 are eligible to enter part 2, which is an open label long-term extension. That means everybody gets drug. So during this, patients will continue to come to the clinic for the assessments, for the treatment period, and for each individual, it'll last approximately 156 weeks. So that means the total length of the study is approximately 4 years. This is a fairly substantial commitment. This is a global study where we intend to evaluate Ion 440 in approximately 48 participants. I mentioned the sham control, so we're sharing with you our randomization scheme. So our randomization scheme is 3-to-1, which means for every 3 patients who receive ION 440, 1 patient will receive the sham control. Now again, that's only for part 1. Everybody who enters part 2 will receive ION 440. And we'll talk a little bit about how that works in the next diagram. Okay. So this is a little bit more detail about the trial design as well as the escalation strategy. So because this is a first-in-human pediatric study and because it is a blinded study, we have implemented an independent safety monitoring committee. I'll use ISMC as an acronym for that. These individuals are experts in MECP2 duplication syndrome and Rett syndrome. They're all physicians, and they are not involved with Ionis, and they are not involved with the trial centers. They truly serve as an independent safety monitoring group. And we have implemented them within this trial to evaluate unblinded safety data throughout the trial. Oh gosh, my time's up. Wow, I just started. We have implemented them throughout the trial, and their job is to make recommendations to the blinded Ionis team and our investigators on whether the trial should proceed as planned, proceed with modifications, or should not proceed. They have a specific subset of data that they will be looking at with each sub-cohort, and will use the information from that data package to make those recommendations. It's only with those recommendations that we can proceed to the next dosing cohort. So let's talk a little bit about how that actually looks and how it's implemented in the trial. All right, so Part 1, dose A. So these individuals all received the lowest dose that we're evaluating in the study, and they completed up to a certain data cut. The ISMC evaluated that data package, and they make a recommendation. They make 2 recommendations. The first recommendation is, should we increase the dose and look at a higher dose in a new group of patients? That would allow us to start cohort 2B. At the same time, they make a recommendation on, can we evaluate that same dose in a younger patient population? And that allows us to start cohort 2A, 1B, excuse me. I'm getting myself confused with my own cohorts. So these 2 decisions have to happen in order to proceed. So what happens then to these individuals who started the study at dose A? They continue. So in the long-term extension, they continue on dose A. At the time that the ISMC makes the next decision regarding the safety of dose B, it's only after that that the participants who were on dose A could potentially receive dose B. And that's really important, and that will happen in an iterative fashion. So what that means is these individuals who start on dose C, these patients will continue, participants will continue on dose B into the long-term extension. At some point, the ISMC will review the data package from dose C. If they consider it to be safe and tolerable, then individuals who were previously on dose A or dose B may have access to dose C. There is an exception to that. Dose C is not available in the United States at this point. It is available in all other regions, so we have very few folks from the US in here. So for everybody in the UK, Europe, other parts of the world can dose escalate to dose C at this point. Okay, so getting away from the design of the trial, I want to spend a little bit of time talking about the trial population. Now, we know that the genetic variant structures for MECP2 duplication syndrome is complex. We've had some very beautiful, elegant work that has been published recently telling us about this genetic complexity. And because of that, we want to make sure that we evaluate ION 440 initially in a patient population that we know more about, individuals that we believe have a putative 2-fold increase in MECP2. And that's why our first study is focused on tandem or interstitial duplication patients. By building a base of information in that patient population, it will allow us to understand how to better dose individuals who have more complex genetic variant structures, including those with triplications. So we build on a foundation that is being established with the trial population in ATTUNE to make sure that we're appropriately dosing everybody with MECP2 duplication syndrome in the future. I am not going to read the key inclusion and exclusion criteria. This information can be found on clinicaltrials.gov. And I just want to highlight, though, that it's just that. It's key inclusion and exclusion criteria. There are others. There's additional inclusion and exclusion criteria that apply. In addition, there are certain tests that happen during the screening process. So it is impossible to know with 100% certainty whether you will qualify for the study or whether a child will qualify for the study until the screening process starts because of these additional tests that have to be performed. Now, a site investigator can feel confident with the available inclusion and exclusion criteria that prior to those tests, an individual may look like a good candidate for the study. But until you actually enter screening, there's no 100% certainty that a participant will screen positive for the study. Okay, so let's talk about the objectives of the ATTUNE study. So this is a safety and tolerability study. So the primary objective is to establish whether Ion 440 is safe and tolerable in individuals with MDS, and we're particularly looking at it in individuals with MDS who have an interstitial duplication. So we do this by monitoring for adverse events. So adverse events are unwanted or unanticipated changes in the individual. We perform neurological exams and physical exams, and we also look at the way that the heart is functioning with an electrocardiogram. This is all standard for a phase 1 trial. In fact, it's standard for all clinical trials. We're going to be looking at these, and because this is a neurological compound and a neurological disease, we include those neurological exams fairly extensively. Now, a fundoscopic exam allows the physician to take advantage of looking at the back of the eye to determine whether or not we have any sort of indications of swelling in the brain that can then manifest by that observation. And this is important not only because it's relevant in the disease, but also because this is an ASO trial. We're introducing a protein into the brain, and we want to ensure that we feel confident that nothing is happening in the brain that we would not expect. MRIs do that even more definitively. So we perform MRIs at baseline and at other points throughout the study, and that allows us to look at the structures of the brain and then to ensure that the structures of the brain are not changing with the introduction of an ASO. Secondarily, we're looking at describing and determining the pharmacokinetics of Ion 440, which is what the body does to Ion 440. We can do that by collecting blood and CSF. So by looking at the amount of Ion 440 in the blood, we can determine what the exposure is and how the body processes Ion 440. This is very important so that we can understand the relationship between the exposure of the drug and the ultimate responses. It also allows us to look at any sort of adverse events that may be happening systemically, because although the drug is administered intrathecally, so through that lumbar puncture, we do still have systemic exposure of the drug. We can also look in the CSF, and the amount of Ion 440 that we can see in the CSF gives us an indication of how much Ion 440 might be in the brain, which is where the target of Ion 440 is. MECP2 is in the brain tissue, and so we can use those pieces of information to better characterize the drug. We also have exploratory objectives in this study, and this is looking at how well does Ion 440 address the signs and symptoms of MECP2 duplication syndrome. On the next slide, we'll be looking at what those signs and symptoms are that we're looking at to determine if Ion 440 has an effect on, but it's important to note that these are exploratory. This study is relatively short. So part 1 of the study, as we mentioned, the treatment and safety follow-up period is only 36 weeks. We follow that with a long-term extension. So some of the changes may happen rapidly. Some of the changes may take longer to manifest. So we're looking at when we start to see a response, if we see a response at different levels, and of course, some of that may take time. Now, we do that by evaluating different assessments. And I'm not going to go through all the details of the assessments, but they're roughly divided into these categories. So ClinROs. ClinROs are clinical observations. So that's something that the clinician or the study staff is evaluating in the clinic. And what that tells you is what the individual can do in the clinic. So it has its limitations. We all know that our children with MECP2 duplication syndrome can have good days and they have bad days. So that's a limitation to the clinical observation. So we also include OBS rows. OBS rows are observational reported outcomes, and they are outcomes that are described by caregivers, parents of individuals with MECP2 duplication syndrome. And they're very valuable because it tells us what the individual can do in their normal environment. So not always when they're at the clinic, but what they can do generally. Both of those pieces of information are incredibly important for understanding, again, if ion 440 affects the signs and symptoms of MDS. We're also looking at seizures with an electronic seizure diary, and we're looking at caregiver burden. So this with self-reported assessments, we'll talk about those in just a second. But all of these help paint a picture of what ion 440 is able to do across the signs and symptoms of this disease. Now, we're going to talk about some of the data that we have from the natural history study. A lot of that informed the decisions that we made and what types of clinical observations and caregiver observations we're looking at. Dr. Pelovin did a nice job of highlighting what some of those are. And Paul's going to spend a bit of time today talking to you about what we found. One of the ones we won't get a chance to talk about, so I'm going to highlight it here, is this caregiver burden. So you saw some of the original published work published by Dr. Pelovin and colleagues, and Ionis conducted a similar survey along with our collaborators at academic institutions to evaluate burden for both the caregiver as well as for the patient. What I like about this survey is it extends that work and it's global. So we have feedback from individuals in the UK, from Europe, New Zealand, Australia, China, and Canada, in addition to the US. So it's our opportunity to collect that feedback and understand what that burden looks like across the entire spectrum and across the globe from the community. Now, I do need to say this, that the evaluation of the safety and efficacy of ion 440 in clinical trials is essential to establish whether ion 440 can help individuals diagnosed with MDS. So for that reason, ion 440 is only available as part of clinical trials at this time. So we all know that MDS is a very heterogeneous disease. So we know that from our research. We know that from Charlie, pictured right here. So a shout out to Charlie and a thank you to his mama for giving us permission to show you this picture of Charlie. And I like this picture because it highlights that not only is it a heterogeneous disease, it changes. The trajectory changes over time. And again, we know that from research, we know that from Charlie, we know that from looking around the room and seeing all of the wonderful kids with MECP2 duplication syndrome that is here today. So because it's so heterogeneous, we are looking at everything. We look at this disease holistically, at all of the features of the disease. This is a huge amount of work. The sheer number of scales in the ATTUNE study is a massive commitment to the families who do participate in any of the research studies. We'll talk a little bit about some of our learnings from NH6 and how that did help us streamline the ATTUNE study, but I will say that this is a huge commitment, but it's also so important to us because what we want to make sure is that a future treatment actually addresses the needs of each individual family, because those needs may change. And one thing we learned from our caregiver burden of disease study is that if you ask a parent of a 4-year-old with MDS, their needs and desires and treatment goals are very different than if you talk to families of a child who's 14 years old, for example. So we want a future treatment to address your individual needs and your individual family needs, recognizing that every journey in MDS is different, distinct, and unique. So here it is, the update on the ATTUNE study. So how are we doing? Cohort 1A is fully enrolled. We could not have done this without the incredible commitment of the families, as well as our site investigators, site staff, and the community who really rallied together to help us enroll this study. Cohort 1B and Cohort 2A are open for enrollment. We have 50% of our sites open, and they're highlighted here. So just a huge thank you to everybody in this room. Many people have contributed. Many people in this room may may contribute even further to our goal of successfully enrolling the ATTUNE study to evaluate Ion 440 in patients with MDS. And I will say, though, that this does reflect and this does represent all of the sites that we anticipate opening for the ATTUNE trial. In the future, we look forward to discussing with all of you other potential locations where we might open up sites, again, for a subsequent trial if the ATTUNE study suggests further development of Ion 440. So I also want to take an opportunity. Maybe I'll go back for just a second. I know there is a lot of enthusiasm about when are we going to share data and results from this trial. As I mentioned, this is a blinded controlled trial. And so right now, we don't know who is on Ion 440 and who received the sham control. That certainly confounds our ability to interpret the data. So our plan right now, and plans change, but our plan right now is that we will not share data from this trial until we have unblinded part 1 of the study. So that should be the expectation, and certainly if that changes, you will hear from our patient advocacy partners who will certainly communicate if anything is disclosed before that, even in a blinded fashion. I pointed to Courtney, she's over there, she's waving. Wonderful. All right, so that's what I can share with you about ATTUNE today, and so I'm going to shift gears. I know, that's all I can share though. I'm going to shift gears and talk about a little bit about our natural history study. So NH0006, which I'm just going to shorten to NH6, is a prospective and retrospective natural history study in individuals with MDS. And the goal of this study was really to evaluate biomarkers in the MDS patient globally, but also specifically looking at individuals who would match the planned ATTUNE population, and looking at longitudinal performance across those CLIN rows and OBS rows that we just reviewed. And doing it at a cadence of frequency that would match what we plan to do in the ATTUNE trial. So I'm very pleased to share that the NH6 study is complete. So we have a sufficient amount of data to fulfill the trial objectives, to do exactly what I just said, which is define the unmet need and disease trajectory in MDS and the ATTUNE population. This is the largest natural history data set for MECP2 duplication syndrome to date. We very much look forward to sharing additional data coming from that study today, and also as we progress towards a publication as the data set gets locked and we can finally share the results of that study. But I'll share a couple of key learnings. So what we learned from this study was that the tests and assessments that we plan to use in the ATTUNE trial were tolerable for families, and we'll talk about the ones that weren't. We removed them. They were repeatable over time, so we could get that nice longitudinal performance, and that they were meaningful for tracking how the conditions change. So we're looking at things that tell us something about severity, and we're looking for scales that match each other, right? It's not helpful if one scale tells you one thing and another scale tells you a totally different picture, because it's not going to be useful for interpreting whether Ion 440 has an effect and improves the signs and symptoms of MDS. You hoped individuals who participated in this study really did help reduce the burden for future families. So we looked at all the assessments, which ones are the most meaningful, and what this meant was we could reduce the number of assessments that we're ultimately going to be looking at in ATTUNE and in future studies. And again, making sure that we have the right tools to characterize these assessments, that we're generating the right data for the future of understanding how Ion 440 works. And then importantly, you helped us identify biomarkers, a word that you've heard a lot today. You will continue to hear this. It's incredibly important that we can measure biomarkers, in part because this is a dose-sensitive gene, in part because we want to make sure that we really understand when something might be changing. What are good predictors of clinical change when we know some of those clinical changes may take time? So this was the direct impact of a lot of what we learned, in addition to characterizing the disease as a whole, is that we were able to change the design of the ATTUNE trial, protocol coming out soon, Dr. Balaban, to improve the participant experience. So what you're looking at here is a complexity assessment. Time in minutes. Looking at those the OBS rows and the CLIN rows, and what we were able to do is use the information we learned from ATTUNE and reduce by 20%, the overall time. Now you can imagine when you're committing 1,200 minutes over the course of the study to these clinical observations, to these caregiver observations, a 20% reduction is hopefully a meaningful reduction in giving your time back to the sites, time back to the individuals themselves, the participants, and time back to the families. And we did that by focusing on scales that were simply not patient first or patient friendly. We didn't have any information leading into the natural history study about what scales might well capture the disease. And so we found out that some of those scales just didn't resonate with caregivers and their families, and they weren't doing a very good job of telling us more about the disease. We got rid of assessments that were not well tolerated. So if they're not well tolerated, they don't generate data that we can use to meaningfully interpret the effect of Ion 440, and that they were redundant. So again, this is a very heterogeneous disease. The scales that we use also evaluate multiple domains of the disease, and so trying to reduce that redundancy wherever possible, but making sure it had no impact on being able to meet the trial objectives of the ATTUNE study. So that's really it for me. In terms of an update, I just need to say a huge thank you to the team at Ionis. There is a huge group behind me and Paul and Courtney that you never get a chance to see. They're working tirelessly on this program and contributed to many of the data and information I shared today. So I do need to thank my colleagues who are not here. I need to thank the community, all of the investigators, and all of the site staff that do this, and particularly the individuals, participants, and their families that contribute to any of our research initiatives, including the trials that I highlighted here today. I need to thank the Global MDS Alliance. You all are amazing. You've brought yourselves together, which is incredible, and I can't wait to see all that you do, and thank you for your support. And last but not least, I'll just thank all of you for your time and attention today. I'll take any questions.
Okay, as you can see from my face, I'm from China. So I'm just curious, will there be a plan for the clinical trial in China or in Asia somewhere?
So right now we are evaluating potential sites in other regions of the world for future trials. So I would love to hear more about your physician and what the site is able to do. So that would be one thing that would be very helpful for us. If you are from a country that wasn't represented in our geographical map of where ATTUNE is, to please come find myself or Paul or Courtney or your local patient advocacy lead to tell them a little bit about the trial and provide us with the contact information. So we are talking to individuals not in the countries pictured here to better understand their capabilities, any sort of limitations with the health authorities, to be able to see where we can bring our trials to in the future.
Okay, thank you. Mm-hmm. And is it possible that we participate in the clinical trial in Europe or in some existing trial?
So cross-border travel is completely acceptable. I think the guidance that we give is, so ultimately the inclusion of trial participants is up to the investigator. So the PI at the site needs to agree to accept a participant who doesn't live in country. And our only guidance to them is that the participant as well as the family needs to speak the local language. So if you're going to, we highlighted that France is open. So if you are going to a site in France, you and your child must speak French. So that's important because the assessments are conducted in the local language and the scales are provided in the local language. So you do have to speak the local language.
All right, thank you.
Yeah.
Sorry. Oh, hello. I'm Veronica. I'm representing Livia and her son. They are from Slovakia, and they are the only one in the country. And as you mentioned, because she's also interested if there will be an international group maybe where she can join with her son. But her son does not speak or understand any other languages than Hungarian, and as they are the only one, they have no chance, I think, to get in the study. Because she and her husband can...
Well, so what we're saying is both need to speak, only because, of course, for scales that require a psychometrist to be able to communicate with the child, you minimally have to be able to understand. Yeah, the child has to be able to understand the commands, so that if they're not doing it, is it because they don't understand or because they're not able to? So what I would say is, our plan in the future is to expand out to other regions in a future trial, but that is the limitation. I certainly, if there is a study center that is nearby, you can contact the PI and understand what options might be available, but it would be up to the PI to determine a path forward if you're not a native speaker of the closest center.
Okay, thank you very much.
Of course.
Hi. So my question was, so you're saying that you're not going to publish any data because you're still in that first phase of the trial.
Yes.
But if you were considering your child for one of the trials, what kind of, it's a big commitment to make without knowing what the side effects are. How do you know if you want to put your child under that risk? It's a really difficult decision to make.
Sure. Yeah. Let me respond, and then I do invite Dr. Pelevin, or actually we have several investigators here who are involved, and they might be able to provide some guidance for you. What I can tell you is, as part of the process of deciding whether or not to participate in the trial, you're given an opportunity to review the informed consent form. Within the informed consent form, we provide information about what we know about the safety and tolerability of Ion 440 from animal models. So that information is available because we have performed studies to evaluate the safety in animal models like cynomolgus monkeys. So I think that is a starting point for being able to make that determination. I invite the investigators to come up if they've got other recommendations for how families might make this very personal decision.
Yeah, I'm running the natural history study. It's over now, and the CS funded the Ion 440. So I was very enthusiastic at the beginning. Oh, I should bring everyone in and everything. But the commitment for the trial is really intense. Okay, I will say that. They're getting the dosing or lumbar puncture every 3 months. That's one. Number two, in each of them, so they get it. Yeah, they wanted to ask. Yeah, so. First, they get it every 3 months, and they undergo sedation. Okay, sedation is not easy for these kids. Okay, this is not because the child won't stay still to do lumbar puncture. Number two, after that, you get a month later, you need to come back to the site again, and on the week of dosing every 3 months, they're here whole week, kind of. Okay, so all I can say is, if you don't have first, the safety of the child, okay? If the child is very sensitive to travel and everything, which most of our kids, I think now I am learning my lessons. I don't want your child to end up in the ICU staying just for the trip. And I know we all want the child to benefit, but the trial, clinical trial means it is a long commitment, requires a lot of effort on the family end. So this is once the trial period is over, because we need a successful trial. Let me say it this way. I may be enthusiastic and I may bring a very sick kid, okay? What if the child ends up in ICU and unfortunately dies? That's gonna scrutinize the entire study. That's gonna hold the study maybe at least years, or the worst case scenario, it's gonna terminate this study. So if we want to bring treatment, we basically, as a scientific clinician, I can say, I want you to be a little bit more patient. Local patients are definitely much preferred, because the lack of the flight, I get tired from every flight. Okay, you can't imagine what these kids will go through, going through the security process and everything. So safety, if the child is tolerating. And number two, huge commitment. If you're a single parent, okay, just even both parents are not enough. You need mostly, I know that for my patients, they other family members needs to support them. if especially both parents are working. So these are very long, like it's literally like too much commitment than I expect, like envisioned. That's all I can say for that part.
Yeah, of course. But to answer your question, there is information provided about the safety that's available in that informed consent. So if safety is your primary concern, then that information, as much as we have, can be made available to you. As I said, right now, our position is that we would like to see the unblinded data before we present it. That doesn't mean that if we feel like there is a demand and a need for us to provide safety information in a blinded fashion, which means, again, it's sharing all of it as opposed to being able to say definitively individuals who this is just the natural history of the disease versus symptoms that may be related to the ion 440, then that's something that could be considered in the future. But as I mentioned, we do have that unblinded independent safety monitoring committee. They are evaluating the data often, and they're looking at the full totality of it, and they do know who is on treatment and who is on sham, and they're making these recommendations to us, and so that's how we can also feel confident. Ionis, I'm blinded, Paul's blinded, Courtney's blinded, so feeling confident that we can continue as well is by relying on these experts. We look at the blinded data all the time. I think Paul goes into EDC every day. And so we are looking, and our medical and safety teams, all physicians, are looking at the data in a blinded fashion to ensure that we're not seeing any trends that would surprise us. We're not seeing anything that's unexpected. And we conducted a natural history study to very much be able to say, is this a natural part of the disease or not? So all of those things are in place at the same time, but I do recognize it is a very challenging decision.
Can I just... Oh, okay. I just had one really quick follow-up question.
Please.
Do you know, many years ago when we were first talked about the trials, they said that the best effect of any possible treatment would be on kids that are younger, because then, you know, their ability to relearn, you know, some of the skills that they haven't learned is better. Like our son's 14 now, you know, by the time that this treatment, if it gets approved, he'll be a lot older. I mean, is there, because I know that the second group is like, what is it, 7 years plus to like 64 or something?
8 to 65, but
Yeah.
yes,
Yeah.
very close.
But obviously then you're considering like people from all age ranges, really.
We are. So again, there are certain stability criteria that are necessary to be involved in the trial. Dr. Pelevin pointed out some of the challenges and instability of our patient population as a whole, but also we do know that trajectory changes as the child gets older. So right now, we have animal model data in both young animals as well as older animals. There does appear to be a value proposition in both. We haven't done an extensive analysis in the older mice, but we do know that from the electrophysiology data that was shared today by Dr. Pelevin, those little squiggly lines, those were from mice that were 25 to 35 weeks of age. So that is an old mouse. And as you could see in the mouse model, it completely abrogates that response. So the goal of this trial is to assess, is there a difference in the safety tolerability or in the potential efficacy of ion 440 in those different populations? And that's part of the reason why we designed the trial that way.
One in the back first, then maybe.
Hello. Hi. Thank you for the presentation. Just a very quick one. My son is going to be 2 next year, so we are kind of really curious about more trial information, and I'm not sure about the timelines of when you might be showing more data about 440. I know you say 42 weeks, but is there some timeline for the coming year, just to have access to more information?
Sure. So I think our timelines are available on clinicaltrials.gov, and so I believe our primary readout 2027. Paul, yeah, okay. Paul's nodding at me. So we believe that we'll have our top line results, which means our initial trial results from part 1 of the study in 2027.
Thank you. Can I ask my question? It's about the places where over here.
Thank you.
No problem. So, Can I ask you please to return to the presentation sheet where it shows the places where it's going to hold the clinical trials? Okay, I can see that most of them are in the States. Seeing, of course, that Ionis is from there, so it has this kind of facilities to have the clinical trials. And then in Europe, there are like 3 places.
4.
4 places. Okay. What was the qualifications for these places?
Sure. So in the United States, we went with centers of excellence. So these are individuals who are experts in MECP2 duplication syndrome. They also, 5 of them participated in the natural history study. So they were already very familiar with the assessments, with the administration, and all of them have previously participated in antisense oligonucleotide trials. So they have the infrastructure for doing this, or they're administering ASO. So some of them are involved in, for example, administering nusinersen to patients with spinal muscular atrophy. So those were a lot of the criteria, was to make sure that they were familiar with the disease, they were familiar with the outcome measures, and their site had the capabilities. The same philosophy was applied to the regions in Europe that we selected, and that was really we know about the patient population in those countries. So the more information we can gather about the population of MDS in individual countries, so this is my plug for REMEDS. If you're not familiar with REMEDS, you will be by the end of today. And I do hope that you consider registering. It's a global registry for MECP2 duplication syndrome. That will help us. So we did focus on regions of Europe where we know that there is a large bolus or group of patients with MDS. We also needed to ensure a same criteria apply. The physicians are experts in the disease or a similar related disease. The site has the infrastructure to accommodate a trial of this nature. And those were really the main criteria that we used to establish that.
Thank you. This leads me to the second question, because meaning about the ballot of people. So in Greece, now we're we have four kids right now. We know for some more. But what kind of qualifications will your company reach out to the physicians in Greece, child neurologists, or other kind of expertise on research for that? Because I'm sorry.
No, please.
As I see, there's UK, there's Spain, France, as you said, Austria is the other country.
Yes.
So, what kind of how did you reach out to find this? Did you had prior expertise? Language barriers.
That's okay. That's okay. Take your time.
Prior communication, cooperation with them, so you know what kind of tests places are there, so they can help you with your research.
Yes, so this is an opportunity where when you're in a rare disease space, me as a clinical researcher, I enter a new space and I look to see who the experts are. And how I do that is I go to this lovely place called PubMed, and that is where all scientific publications are listed. And if I look at who is publishing, who is writing articles about patients with MECP2 duplication syndrome, I can get an idea of who sees these patients, who knows these patients. That's helpful for me, so that helps me. But I would say in this disease, we had friends. So we had friends at Baylor, and they introduced us to other friends. And that's how our network grew, and we got to meet these amazing experts. Then the community started finding us more experts too. So as we engaged with the community, they helped us find new people who we didn't know and we didn't have in our network. And that's how we've been able to grow. So for example, in Greece, we're talking to the investigators in Greece, because the minute we get a recommendation of somebody to reach out to, we reach out to them. So I'm talking to investigators in Japan. We're talking to all sorts of groups who are interested in, one, understanding more about where we're at now, what we're doing, and what does a potential collaboration look like. So the request is, please keep telling us about your physician or physicians that you know who sees these patients and sees your family members, and that will help us establish that broader network for the next step.
Thank you. As you told me about REMEDS, I've sent the DNA and stuff years ago, so I'm familiar with that. And the other thing is that, as you said, that we're friends, and through friends you found out.
Yes.
We do have some doctors that we can talk to and reach out, and we know. So if there's a way that we can communicate on that,
Yes.
since there's a lot of, let's do that.
Absolutely.
And thank you very much for my time.
Okay, and what I would say is after we finish these questions, I would love to invite somebody from one of our patient advocacy groups to kind of come up and talk about that, or if we don't have time, where's Helen? If we don't have time for that, then maybe in the next section where we talk about that, it'd be really helpful to provide that clear mechanism for everybody in this room for who you should reach out to help start that conversation. Oh, do you want to do it? There we go. Just come on out. Yeah.
Yeah, so I just want to clarify a little bit, but you will hear this a little bit later. There are different registries. Maybe also there was the genetic studies that was done a couple of years ago. HEMETS is the new registry. I will talk about it more in the afternoon, so you will hear. And I strongly invite everybody to register on HEMETS, because this will also help identifying where families are and identifying where centers of excellence could be. We are in communication also with Ionis team, with also representative of families in different countries. For Europe, it's a little bit difficult because there is a barrier language, unfortunately. We are also insisting on Ionis to include translators in the study. I know this is something that is in the mind of Ionis. And what I want to say is, as soon as you know you have an expert, you are confident that your doctor can conduct a clinical trial, send us an email either to one of the representatives of the Global Alliance. We are here to transmit this message to Courtney, to Christine, to Ionis team to discuss it, so that they can look into this center potential and start the conversation for future trials. So that's very important. As soon as you have a contact, come to us, come to one of us, and we will share this information.
Sorry, it might be more than one question. There's a couple of things. So when you said the anticipated top-line results will be 2027, is that different to the unblinding stage, or will that be earlier?
Sure. So the order of operations is that we complete enrollment, and patients complete that 36 weeks in part 1 of the study. At that point, there is a process where we ensure the completeness of the data. So the data is being captured in an electronic data capture system. We ensure the completeness of that data. Then we lock it, and that database lock just means we can't change it, right? So we don't change the data at that point. At that point, I fill out a little form, and my statistician does, and we unblind the study, which gives us the unblinding treatment codes, which exist, but they're not accessible to anybody on the blinded team. Shortly after that, once we've unblinded the study, we would have the initial results internally at Ionis. We would review those results. We would review them with the investigators and determine what our interpretation is of those results. And so, traditionally at our organization, that would lead to a press release with the information from the trial, and so that's the reference to top-line results.
Okay. The next question would be: is the French study already fully enrolled? But probably related to that, and I don't want to jump the gun because I see there's a presentation about Birmingham. Is the Birmingham study one of these, or is it a different?
So it's all one study. So the ATTUNE trial is one study with global sites. So the sites here in pink are sites who are able to actively enroll, meaning they're recruiting and accepting potential participants. The site in blue, there's still a few more pieces that are needed before they can start accepting participants. No, the study is not fully enrolled because of that stepwise process. So cohort 1A is fully enrolled. Those 8 individuals have entered the study and gotten to the next stage. And then we have 2 cohorts that are actively enrolling. Each of those cohorts is 8 individuals. So right now we're recruiting 16 individuals. After that, we'll recruit an additional 16 more, and then the final cohort is 8. So that's where we're at.
Okay, thank you.
So one question about ages. So for B cohort, it only goes down to 2. How do you guys generalize that in the future for like the younger kids if this does get approved?
So we are actively evaluating the feasibility of looking at Ion 440 across different genetic variants and obviously across different age ranges. One of the challenges is the delayed diagnosis of MECP2 duplication syndrome. I'm sure many of you are familiar with the delayed diagnosis. So we are actively tracking with partnerships with patient advocacy, any family who is engaging with patient advocacy after they receive that diagnosis to understand how many individuals, how many infants we would have to potentially evaluate Ion 440 in. There's also other mechanisms that you can use to determine what the appropriate dose would be in an infant, so we can use extrapolation models, but ideally we would empirically evaluate Ion 440 at minimally the safety and tolerability as well as the PK profile in an infant population, and that's how you could move forward with having the ability to treat infants.
Okay, thank you, Christine. It's really helpful. Thank you, and thank you everyone for your questions as well. We'll take a quick 10-minute break for a coffee. If we can come back in at 25 past, that'd be lovely. Thank you.